Okay, thank you very much for the invitation and for the introduction. It's a pity that I can't be in Vienna myself. Originally, we planned doing that. So I spent a very nice time, 3 months or so, in Vienna when I participated in the so-called Young Summer Scientist project at Luxembourg, and I really enjoyed being there. And I've visited Vienna several times since then, and always liked the environment. But of course, that's not possible at this moment, and I've now to talk to a computer screen. I hope that nevertheless it will be somewhat enjoyable what I have to talk about. Now, my talk will be about a mechanistic perspective on the evolution of adaptive plasticity. And to give it a start, I would like to start with this quote that is often repeated in evolutionary biology: nothing in biology makes sense except in the light of evolution. Now, if I use this quote among evolutionary biologists, everybody is nodding and saying, okay, that's great. But if I talk to other people, so like biochemists or neurobiologists or medical biologists, then they typically say, no, this is really quite an arrogant kind of statement, because the work that we are doing is just not using evolutionary thinking at all. And for example, my wife is developing vaccines and she's a virologist. So in her daily work, she does never really make use of evolutionary insights. And I think part of this is is related to the fact that biology is nowadays, or the life sciences are really divided into very different branches. On the one hand, you have the proximate life sciences who strive to unravel mechanisms that are underlying organismal behavior. So here I've shown a brain, so more or less how the brain is working, or molecular biologists are interested in how metabolic pathways are working or something like that. And on the other hand, we have then the ultimate life sciences like ecology and evolution, who then focus on processes that are very complicated, like natural selection, if you think about it, but who largely neglect mechanisms in their work. And I personally am very much convinced that in a longer term perspective, we have to reunite the two kinds of ways of thinking in order to introduce the more evolutionary thinking into the proximate life sciences and introducing more mechanistic thinking into evolutionary biology. Now, there are, of course, now some tendencies towards such a reintegration. So, for example, here, this is one of the basal books about Evo Devo, where a field which is then more or less trying then to merge evolutionary thinking into developmental biology and developmental thinking into evolution. Or there are some attempts towards evolutionary medicine where people then try to introduce evolutionary thinking into the understanding of disease, and on the other hand, also have produced quite some knowledge that is evolutionarily quite interesting and important. A third direction is evolutionary systems biology. Systems biology is more or less on the unraveling of, for example, metabolic pathways and so on. And there are also some people who are trying then to give an evolutionary touch to this. So there are some examples where evolutionary principles start to play a small role in the proximate sciences. However, this has not really gone very far until now. However, there is still some hope because, and mainly I think the hope has to do with the fact that nowadays all of us are using molecular techniques, and we apply them routinely, not only in the proximate life sciences, but also in modern ecology and evolution. And I think this will also lead to a kind of reunification, just because people are using similar techniques and also are using the same kind of language. But for the talk that I want to give now, one of the main questions is now, if we as evolutionary biologists, if we introduce mechanistic thinking into our work, does it really matter for our predictions, for our view of how evolution might work? And there are some examples that clearly show that mechanistic thinking may make quite of a difference. So, for example, there's been a lot of debate about the relationship between robustness and evolvability. So in former times, people always thought now a system is either very robust, that means is not easily affected by mutations or all kinds of perturbances, and evolvability, which means that the system can evolve very rapidly to new environmental challenges. So people always thought there is a trade-off between robustness and evolvability. A system that is robust is not evolvable, and a system that's very evolvable is not robust. However, if you look at the mechanistic perspective, as for example has been done by Andreas Wagner and his group, then basically the situation looks very different. Andreas Wagner has looked at the evolution of large gene regulatory networks, as you find them in many bacteria, for example. And what he found there is that if you look at the evolution of these regulatory networks, so these mechanisms underlying then responses to environmental conditions, so if you look at these networks, then it turned out that robustness and evolvability are two sides of the same coin. Basically, what he found out is that if you let them evolve, then those networks who show the highest degree of robustness also show the highest degree of evolvability. In other words, the mechanistic perspective leads to very different predictions than the classical predictions that you find in old textbooks in our field. The problem here is that Andreas Wagner's work is very, very nice. I like it very much. However, it is based on a very simplistic fitness concept. Typically, he looks at networks that only have fitness 0 or fitness of 1 and nothing in between. And he also does not include any ecology there. So basically, fitness falls from the air, and it's not that clear how ecologically relevant these findings are. A second example about mechanisms, whether mechanisms matter. And this is from the work of the group of Richard Watson from Southampton. And basically, he also looked at the evolution of regulatory networks. And basically, what they found in their simulations is that evolutionary networks, if they evolve, that they learn to generalize over environments, and that they lead them to response patterns in organisms that leads to adaptive performance of these organisms in environments that their ancestors never encountered before in evolutionary history. Our classical textbook knowledge is always saying now that we can be adapted to something that has been encountered before by our ancestors. But apparently, if you look at these regulatory networks, this is not necessarily the case. And therefore, it's also quite a clear-cut deviation from the textbook knowledge that we learned, I at least learned, at the university. But again, there is still a problem there because the work of Richard Watson, he comes more from the artificial intelligence area. He uses quite artificial assumptions that most biologists would have problems to accept, and therefore it's not that clear to what extent these ideas are really also relevant in biological systems. So my own work is, I really want to start a bit more from an ecological setting. So I really would like to look at a mechanism-based approach, but which is then very deeply rooted also in the ecology of organisms, and not that much based then on artificial assumptions on phenotypes and fitness and so on. Moreover, I've mainly focused then not that much on fixed phenotypes, so something like structural genes that affect then a certain kind of the phenotype in a very particular kind of manner, but I'm very much more interested in plastic responses to environments and so on. So something like regulatory pathways, like these gene regulatory networks and so on. And I personally think that this is a field that has not been given sufficient emphasis in the past. So if you look at estimates about how many genes in many different kinds of organisms are involved in regulatory processes, then you typically end up with estimates around 50 to 70% of all genes being then more involved in plasticity rather than in fixed phenotypes. So the things I think, looking then more at plastic responses is in my view something that may be quite important and should be done more often. And as I said, I would like then to switch to a more mechanistic perspective. What do I mean by that? Now, basically, if you look at plasticity, then the traditional perspective is more that one looked at the evolution of so-called norms of reaction. So basically, one looked at something like here we have different environmental differences, and then here we have a certain phenotype. Basically, for example, if you have under cold conditions, you produce a phenotype like that, and under warm conditions, you produce a phenotype like that. And then you have a certain kind of curve that describes then how for a given genotype, then there's the phenotype that is expressed in relation to the environment. And these so-called norms of reactions, they were viewed then as the targets of selection. Now, I try to use a slightly different perspective, and what I call then more the mechanistic perspective, because I do not really think that something like this norm of reaction is in any way then realized in our genome. I don't think that this is the way how responsive or how regulatory processes work. In reality, these kinds of norms of reactions, they result from some regulatory processes which often take the form of a kind of a network that process some environmental cues, and which then more or less then transmit some information about these environmental cues and then produce some responses. And basically, the selection is not that much on the norm of reaction directly, but it is more on the inner workings of these mechanisms. And so basically, the target of selection is the mechanism, and only secondarily then this produce then the norms of reactions, which are then in the end determine then fitness. And I hope to convince you that such a change in perspective really makes a difference and leads to different kinds of conclusions. Now, to briefly summarize, so our approach is then basically include a mechanistic perspective, but with a strong focus on the ecological setting. And we look at highly simplified mechanisms, regulatory networks, to keep our models manageable. But still, our models that we are looking at are so complicated that it would be completely impossible to analyze them mathematically. I'm a mathematician myself, but I think it's just impossible. So basically, what we need to do is really base our conclusions on individual-based simulations, as I show somewhat later. And the main question that we ask is then, do mechanisms matter for the cause and outcome of eco-evolutionary processes? And I want to discuss this in two case studies in order to give you an idea on how we are working, how we are proceeding, in order to give you an idea on how a mechanistic way of thinking might really be an interesting change in perspective. Now, the first case study is based on the work of my former PhD student, Jordi van Gestel. I will show you a picture in a moment. And he studied biofilm formation in Bacillus aptilis. And here you see something like a biofilm. This is a colony of bacteria, which then first grow, but then as conditions become difficult for them, for example, if the nutrients are exhausted or other things happen, so then they have to decide on whether to continue growing and dividing or whether to sporulate. And this is a very important decision that the bacterium needs to make. Being a spore means that you cannot grow and divide any further. So sporulation, if you sporulate too early, basically you do not, you more or less, you give up a lot of offspring production that you otherwise could do. On the other hand, as a spore, you are protected against then adverse environmental conditions. And if you try to sporulate too late, then you may not have sufficient energy to complete the sporulation process. So the adequate timing of sporulation is really a life-changing event then of bacteria. And the question is then, how should bacteria make their sporulation decisions, dependent then on the local conditions that they perceive? That's the question that we asked ourselves. And a lot of research has been done there. And here's Jordi, who is at this moment postdoc in Andreas Wagner's lab in Zurich. And he has then studied biofilms then experimentally, but also by means of a theoretical model. And the theoretical model looks a little bit like this. So we have something like a plane. So we have a planet dimension where the biofilm is growing. And basically blue you see here then are cells, bacterial cells that can divide. And by dividing then more or less are shifted more or less, the biofilm is then expanding in the area. Here you see the level of greenness indicates then the nutrient concentration. What you see here is that light green means that around the biofilm nutrients are getting exhausted, and outside you have then plenty of nutrients there. And basically what can happen in this model is that the bacteria, they can sense their own energy status. They can also sense the nutrient concentration, and they can also sense something that we call signal, which is basically an indicator of the local cell density, say. And this is the model that we looked at, and in this kind of context, we then investigated when then this bacteria should sporulate. And here the spores, they are given in red, and basically these spores are the carriers of fitness, because new biofilms are then being produced by spores and not by growing cells, which could not survive the adverse conditions in a nutrient-poor kind of environment. Okay, so now we look at the two different kinds of perspectives on making the decision on when to sporulate or not. And I'll show them, go into this in detail in a moment. But basically, on the one hand, we take more the classical reaction norm perspective, and we contrast it with the mechanistic gene regulatory perspective. And so in the first, the selection shapes these norms of reaction, and the second selection shapes then the underlying gene regulatory network. And we'll then ask, to what extent do both types of model with a different kind of perspective lead to different kinds of conclusions and predictions? So let's first take the reaction norm perspective, these evolving reaction norms. Now in this case, basically, this is something perhaps important for you to realize. In Vienna, you are working then in a population genetics environment, where one typically starts at the genome level. One looks at all kinds of genetic regions, or looks at gene loci and so on, and then based from the genetic level, then one works up to the phenotype and makes all kinds of assumptions on fitness and things like that. We typically do a different kind of approach. We start more from the ecological challenges that are out there, and then we are looking then more or less how these challenges can be met by some phenotypic adaptations, and then we imagine more or less some gene loci that might then be responsible for the phenotype. And this is a kind of a complementary kind of approach. The hope is, of course, that in the end both approaches will meet somewhere, but you should always keep in mind so that the approach that I'm presenting here is quite complementary to the approach that you will probably take typically. Now, how does our approach then work? So, for example, in our case here, we have then each cell then gets some information about local density, about local nutrient concentration, and about the energy level. And basically, what we assume is that the cell will sporulate under certain conditions, namely if there are too few nutrients there, and if there is insufficient energy, or under some density configuration or something like this. But basically we assume that it is given by so-called norm of reaction, which in our case is represented by this plane. And basically it means that if the conditions of nutrients, density and energy are above this plane, the cell will continue to grow. If you are below this plane, the cell will then sporulate. That's basically the idea. Now, how can you then, how can this plane be modeled? Now, mathematically speaking, it's quite clear. Such a plane is given basically by three values giving something of a certain weight to the nutrients, a certain weight to the density, a certain weight to the energy, and a certain threshold value. And what we assume then is that these weighing factors, alpha n, alpha d, and alpha e, and the threshold theta, that these are genetically determined parameters, which determine then this norm of reaction. And then these are, in our case, for this evolving reaction norm model, these are then the evolving entities. So the putative gene loci that more or less then evolve, and with this change in these parameters, also the norms of reactions evolve. And then we can just see how do these parameters evolve in replicate simulations. So there's nothing new. Many people have done something similar before us. And basically what happens then in our case, now first we start in generation 0, where there is only something like a nonsensical norm of reaction. And basically what happens here is that we have a biofilm where there are hardly any spores. Only once in a while you see a certain spore here due to a mutation or something like that. But basically this biofilm is not very productive. But very soon you see that over a few generations, so say for example 100 to 200 generations of biofilm formation, you see that there is then the production of quite a high number of spores per biofilm. And the productive biofilms look somewhat like this, that in the fringes you have then growth by bacteria, and in the center you have then spores who are formed in that part of the biofilm where the nutrients have to have been exploited. Basically what you see here is, this is the outcome of 500 replicate simulations. And one interesting thing is that the outcome is not really unique. So you see that in all the simulations, they end up in a broad spectrum of spore formation. Some of them, they manage to some of these replicates end up in a state where the biofilms produce, say, 1200 spores. In others, only 900 spores, and still others, you get something like 500 spores. You see something like considerable variation in outcomes of evolution. And if you try then to look how these outcomes look like, now then it looks, we have here then represented them in so-called phenogram. And basically you see that there are actually 1, 2, 3, 4 different outcomes and another outcome which is called F here. And basically what you see is that 4 different norms of reactions have evolved, or actually 5 different norms of reactions have evolved in these 500 replicate simulations. Some of them produce these kinds of biofilms, others these kinds of biofilms, others, and you see different types of biofilms. And only those in this part F, you see they produce these biofilms that also look like real biofilms of Bacillus subtilis with the spores in the middle, and then a broad fringe where still growth is taking place. The first question is, of course, now why have then some of these simulations ended up in a reaction norm that was not very is not very adaptive or does not yield a very high fitness, because as I told you before, spore formation really corresponds to the fitness of the biofilm in the end, and so high degree of spore formation is really very important for them. But I think what happens there, and I could tell you in more detail if you would be interested, so that basically what happens is that all simulations start somewhere here. And what happens is basically that they evolve into different kinds of directions and then end up in different local fitness peaks. And only one part of the only small subset of all simulations end up in this particular fitness peak, which corresponds to this norm of reaction, and which then produces a very high number of spores. So basically what we see is then there are many different outcomes. So we get many different outcomes of the replicate simulations, but all the outcomes that have a very high number of spore formation, they look actually very similar. So these are, for example, the top 20 simulations there, and all of them produce a very similar norm of reaction and also a very similar kind of biofilm. So this is more or less then a summary of the more traditional way of looking at the sporulation process. Now let's switch a little bit more to a mechanistic way of looking at it. Now the sporulation decision is very well known in Bacillus subtilis, and basically here you see a very highly simplified version of the regulatory networks that you find in these cells, where many important decisions are being taken. Basically, you see that some information is coming in via receptors, and this information is then processed, and in the end, here a sporulation decision is being taken in this way. So at first, we started to really implement the whole network and make the very, very complicated model out of it. And we got some interesting results, but it was very, very difficult to see the forest through the trees. And therefore, we decided to take a much more simpler approach first, to get a more qualitative conceptual idea of what such a mechanistic approach entails. And basically what we assumed is that we actually have only then 3 different receptors. One receptor more or less having as an input the local energy level of the cell. The other is then the local density, and the third is the nutrient. And then we have so-called a kind of regulatory network, that a layer that corresponds then to the production of some transmission factors. And then in the end, the decision is being taken on whether sporulation occurs, yes or no. Basically, what happens now is that here you have then 3 inputs. And so here you have the connections that are here shown in green and red. These are the evolvable properties now. Basically, what you see here is that in principle, each of the receptors is then linked to all the 3 regulatory layer properties. Some of these connections then evolve to 0. And some of these evolve to positive values, so there's some kind of activation happening. And some of them evolve to negative values corresponding to some repression. Sorry. And, and some of these interactions are quite weak and others are much stronger. And basically what you here see that all these lines, they correspond then to a certain state of the network. And these different parameters, they are then, these interaction parameters for each of them, we then again assume that there is a putative gene underlying it. And these genes, they can then evolve over the generations. Now the question is then, what happens now if we look at the evolution, not of the parameters of the reaction norms, but of these parameters of the gene regulatory network. Now, first of all, when we look again at evolution, then again we start at somewhere where hardly any spores are being produced, and then also within a short period of time, we also get then the evolution of quite a high level of sporulation. But if you compare the two, you see, okay, the gene regulatory network, again, we have 500 replicates. You also see quite a large variation. You also see that there are few replicates that did not evolve efficient sporulation at all. The variation that you see here is at a somewhat higher level. So you see the scale is somewhat different. Here the scale goes to 1500 and here it is above 1500. So basically on average, these gene regulatory networks are performing a little bit better. But if you just look at these graphs, there is not much of a difference. However, it turns out that the outcome is quite different if you again look at these phenograms. Basically, what you now see is that again you get all kinds of different evolution outcomes. So again, you see there many different potential outcomes of evolution in terms of these different kinds of parameters. But the interesting thing is now that here in green you see the 20 best performing replicates in our simulations. And while these best performing replicates were all clustered in one spot in the reaction norm kind of graph, you now see them all over the place. So here is a very well-performing one, here's a well-performing one, here a well-performing one. So in all the branching, you see then really highly performing, say, gene regulatory networks. So you see huge differences, but also huge differences among these well-adapted gene regulatory networks. And this is something that is quite different from what we saw before. Now, if I now just look at how the biofilms look like of the top 20 gene regulatory networks that have been produced, then these biofilms, they look very similar. So you see that at the biofilm level, you see hardly any difference. If you then look at the induced reaction norms of these general regulatory networks, then you see that there are quite some differences in how these reaction norms look like, and quite surprisingly in my view. And if you look at the evolved networks themselves, then it's quite amazing, at least if you haven't thought too much about it, how different the networks look and are, despite of the fact that they all produce more or less the same biofilm with the same productivity, with the same fitness. So you see that some of these gene regulator networks are very tightly interacting and linked with each other. Here you have a very small linkage. You have the sign of the interactions. For example, if you compare these two, here you have some positive association. Here you have negative associations, and so on. So I find it quite amazing if I look at this, so that the outcome of evolution can be at the network level, and therefore also at the genetic level, can be extremely diverse, despite of the fact that they show a very similar phenotypic behavior. So you have a huge diversity in networks, and all of them have a similar phenotype in terms of the appearance of the biofilm and very similar fitness. One other interesting thing is that because very different networks can produce very similar kinds of phenotypes, it is also possible that many of these gene regulatory networks can coexist with each other. And that's something that you also see here in the course of evolution. We always looked how many different networks did we find in the population. And so most of the time, one of the networks was somewhat dominant. But even after a long period of evolution, we always found periods where you had a large degree of diversity across the networks. And so this is also something, in my view, that is quite important, because what we see here is, since very different networks can produce very similar phenotypes, they can stably coexist for a long period of time because of their phenotypic similarity and their same fitness. And this is a variation that is clearly there, but which is cryptic within the populations, and which is not directly apparent if you just look at the phenotype. Final point that I want to make in this case study is, now we look then what happens now if we take then these biofilms that all were involved in one particular kind of environment, and if we expose these regulatory networks to different types of environment. Basically what we did here is we created something like 150 different kinds of environments, and then we exposed the 20 best performing reaction norm networks, reaction norm biofilms, and we put also in there the 20 best performing gene regulatory networks. And then we use them kind of a classification scheme in order then to classify the different networks and the different kinds of environments as to their similarity. The first thing that we see here is that all the reaction norms, they cluster together. So we have not really imposed this. This is basically the result of the analysis, was that all the reaction norm biofilms, they all clustered together, and all the gene regulatory networks clustered together. And if we now look at the two types of clusters independently, then the first thing that we see here, that is for the reaction norm, for the reaction norms, if you expose them to different environments, you see very clearly vertical stripes. Basically, what that means is there are some environments which are favorable for sporulation, and basically all the different genotypes that we have here all perform here very well under these environments. There are other environments which are quite adverse, and then all the networks that evolved, all the reaction norms that evolved are basically performing really badly. So you see then something like these red stripes. The situation is completely different if you look at the evolved gene regulatory networks. There you see some areas like here where basically then these networks that you see here, all the lower networks, they perform very badly under these kinds of environments. So that's one thing that we see. We also see that there are some combinations. So basically these networks, they perform rather well in these kinds of environments, but you do not see this clearly vertical stripe pattern that you saw before. In contrast, you see some networks, they are performing uniformly quite badly when being exposed to different environments. And some of them, they are performing very, very well, irrespective of the environment. So some of them are, in a sense, pre-adapted to all kinds of environmental changes, whereas others are very badly coping with changes in the environment. So a very different kinds of situation than we had in this in the reaction norms. So basically, the conclusions that I would like to see is that in a given environment, many different networks can coexist for long periods of time. So basically, because these different networks, they have despite of the fact that they are quite different genetically and also as networks, so they have the same phenotype, induce the same phenotype and the same fitness. So they basically can coexist with each other. When conditions are changing, this can be very important, as we all know, so that in order then to adapt to novel conditions, now then we need variation, and basically this variation gets exposed to selection, and this allows then a very rapid adaptation to novel conditions. So if we more or less look at the same kind of simulations under fluctuating conditions, then the gene regulatory networks, they can evolve much faster than the reaction norm kinds of networks. And then the other thing that I would like to make is that these networks that are evolved under slightly fluctuating conditions, so they start then to perform well on a broad spectrum of conditions. And a very interesting finding that we repeated more or less what also has been found by others, among others Andreas Wagner, is that if you look at the genotype-phenotype mapping that these networks then produce, that the adaptive mutations occur more often than maladaptive mutations. So basically, based on the phenotype on fitness, so basically we get something like a bias in mutations towards mutations that have a higher fitness than we originally had. So in other words, all this taken together, we arrive at the conclusion so that these gene regulatory networks, so more the mechanistic perspective of evolution, they adapt much faster than the reaction norms, and therefore, which may be quite important if we then consider changing environmental conditions. So this is basically the first case study. Now I would like to switch to a different case study, which is completely different, but still shows very similar kinds of principles, and that's the reason why I want to show them both. So in the second case study, we looked at predator-prey coevolution. So as I said before, we always start from an ecological problem. And then we, starting from the ecology, we work our way back to potential mechanisms and then also to potential genetic architectures that may be responsible for these mechanisms. Now, if we look at predator-prey coevolution, so then, of course, the predators and prey, they adapt to each other. And of course, this evolution is antagonistic. So everything that's good for the predator is bad for the prey and vice versa. The classical approach that people use nowadays almost universally in these models is that one looks at matching strategies. Basically, one assumes that there is a one-dimensional prey escape strategy and a one-dimensional predator attack strategy. And basically, if one looks at the difference between X and Y, then for the predator, it's best to match the prey. So basically, if the predator's Y value is equal to the X value of the prey, now then the catching chance of the predator is highest, catching success is highest. Whereas for the prey, it is best to be as far away from the Y of the predator. Basically, if X minus Y is very small, then the survival probability of the prey is very low. But if they have very different kinds of strategies, then they are very far apart from each other. So this would be the classical way of how these kinds of antagonistic coevolution is often being modeled. And basically, X and Y, they are then encoded as on a single gene loci with infinitely many alleles, or you can also have an infinite alleles model with small effect sizes. But in any way, you can implement this X and this Y and then look at their coevolution. And if you look at the coevolution, then this can often be classified in a relatively simple way. So either the system goes to a kind of an equilibrium, or you get then regular cycles, and nothing really exciting is happening in most of these models. Now we wanted to make this a little bit more mechanistic. And with we, I mean my current PhD student, Christoph Netz, and our programmer, Hanno Hildenbrand. And basically what we did, we looked then at a spatial grid. So basically we looked at a spatial landscape. And on this landscape, grass is growing, and grass is also being consumed by herbivores, so the prey. So you have some individuals, herbivores running around, walking around, and then we have some predators. And these predators, of course, are then looking for these predators, for the herbivores. And then they of course, their fitness then depends then on how many herbivores they consume. Whereas for the herbivores, of course, the evolutionary task is avoid the predators and eat as much grass as possible. Now, we have then, and basically what is now evolving are then in our model movement strategies. So basically we assume now not that the gene regulatory networks determines the movement, but we now think that very simple brain modules or so, neural networks, that they are evolving, and that they basically, these neural networks determine how the herbivores and the predators move in space. And basically it works like this. So take, for example, herbivore. Each herbivore at each point in time, the herbivore looks around and then more or less looks at how suitable are these different habitat spots where I am and in my environment. And then it goes in the direction where the suitability is highest. But what is suitable and what is not suitable, that is an evolving strategy. So in the most simple way, we look at something like this, that the suitability is given then by the weighing factors Wg for grass, the weighing factor for Wh for herbivore density, the Wp for predator density. So basically the suitability is just given by this weighted average of the grass density, the herbivore density, and the predator density. And these are the evolving parameters that then change in the course of the generations. Again, these are then these kind of imaginary gene loci where evolution takes place. But we also look at more complicating decision processes, and actually this decision architecture can also evolve as well. So we can have grass density, herbivore density, predator density, then entering a processing unit, and in the end you get then something like the suitability. Now, I've not much time left, and therefore I would keep it short. Briefly, what we then see is a landscape, and in the landscape you see somewhere then these red dots, which are the predators. You see then some blue dots where there are the herbivores, and green is the grass. And actually we can then run some simulations. I don't know whether it works. Oops. It doesn't matter, so I could show you some pictures. But basically what you see then is that you get a rich change of a succession of ecological patterns. You get something that is often called a spiral wave. You get some rolls, so something like in the Serengeti, where then you have then a movement of herbivores in one direction, followed by the prey. You have some patterns where you have then small scale condensation of predators and so on. And all this is highly dynamic. So you don't have a single kind of ecological pattern, but actually what happens now is that for a while you have, for example, these spiral waves, but then an evolutionary innovation comes about, and then the whole ecological pattern changes quite dramatically, and you have never-ending evolution. It always leads to different kinds of patterns that look very different in the course of time. It's a pity that I cannot show you one of the simulations. If you look, for example, now at these ecological characteristics like predator density and prey density, then you see a pattern like this, that for long periods of time, you have something like a fixed average density of prey, a fixed average density of predators, but once in a while, you see, due to evolution, so some innovation coming about, and then you get here some oscillations going on. Sometimes you also have a certain drop in density or an increase in density. And sometimes here you even have then extinction, and in our simulations we have then re-established the predator. But you see something like looking a little bit like punctuated equilibria. One very interesting thing is that what we found is universally in all of our simulations is that we did not find one type of predator strategy and one type of herbivore strategy. I cannot go into detail here in any way, but basically here what you see here is then at one of these gene loci. In this case, it is the gene locus determining the weight that the herbivore gives to the predators. Basically, it shows then the allelic values that you find in the population. And at first, everything starts at 0, but then what you see in all simulations that we have is that we have then one branch of the population where this allele becomes predominant, which is negative, meaning that these herbivores give a negative weight to predators, meaning that they try to avoid predators. But in all the simulations, we also find another branch where then part of the herbivores actually are seeking the spots where the predators have been. And the reason behind this is that they anticipate that the herbivores, the predators, will move away from the high density spots that they have been before. And this is what you see here. If you look then more how the predators have evolved, and again, you also see that there is polymorphism in the population. And some of these predators have a very strong tendency to avoid each other. So it's kind of a density competition avoidance effect in a sense. And this is basically where these guys are profit from. But on the other hand, there are some predators who do not have such a strong effect, and they are more or less waiting for the predators to appear where they have been at high densities. I cannot go into much detail, but the point is, what we see is that something like polymorphisms and unexpected strategies is something that is then the rule rather than the exception in the simulations. One other thing is that we also see is that if you look at different replicates, that the general pattern is quite repeatable. However, we always get some very different kinds of attractors there. So very alternative outcomes, a few alternative outcomes of the replicate simulations. And here I just show this by the information use of the herbivores and the information use of the predators. And basically what you see here is, interestingly, the predators always have a higher evaluation for the grass than the herbivores in all cases. But here, for example, you see in the first replicate, the herbivores have a very strong tendency to make their behavior dependent then on the predators. Whereas here in the second simulation, they have a high tendency to making it dependent on their own kind. So basically, we see that there are alternative outcomes of the simulations. Now I'm at the end of my talk, and basically the conclusions of this case study number 2 is so that again what we see is that if you have these mechanisms evolving, that we get very rapid evolution, and this rapid evolution it's really orders of magnitude faster than the evolution in the standard ecological models, and this means that evolution is acting on a similar time scale as the ecological processes, and therefore we get a strong interaction of ecological and evolutionary dynamics. The other thing that we saw is that we have a long periods of stasis, but also never-ending change, so never ever really locks into one particular kind of outcome. And these regulatory networks, they allow for really innovative solutions that surprise you when you first look at it, like the herbivores were seeking places with a high predator densities on the previous day, something like that. But if you think a little bit about it, then these solutions make perfect sense. And the last thing that I want to say is that as in the previous model also with these gene regulatory networks, you get very often coexistence of alternative strategies so for extended periods of time. Now, the general conclusion that I would like to draw is that the evolutionary predictions based on mechanistic models, they can be quite different from those of phenomenological models. I think that's the first conclusion that I hope that I've convinced you about this. And the second thing is most of the models that theoretical biologists are making are of the toy model kind. So toy model kind. So you try to make the assumptions as simple as possible, and in order to be able to draw any analytical conclusions. And I have done so throughout my whole career. I always tried then to keep the models analytically tractable. Now that I'm now confronted with much more complicated kinds of models, I'm forced also to make use of individual-based simulations, and then I see that the models with this based on these individual-based simulations, which have more degrees of freedom, can lead to very different predictions. And I don't think that Albert Einstein has ever said this, but still he's often then said to have said models should be as simple as possible, but not simpler. And that's basically also the kind of quote that I always use then at the end of my courses of theoretical on theoretical modeling. So that was more or less what I wanted to say. Thank you very much for listening to this. And I would like to say at the end, so you may be disappointed how little genetics was in this talk, but I think the interesting thing is perhaps so that we try then more or less to approach adaptive evolution from very different directions. I come more from ecology, where the interactions of the individuals more or less automatically lead to fitness consequences. So I don't have then to postulate fitness functions or fitness coefficients and things like this. They are more or less automatically there. But I have a very naive picture then of the underlying genetics. And you guys typically start with a very complex idea about genetics, but then you make very simplifying assumptions about selection coefficients, and you neglect then often frequency-dependent selection and how fitness is emerging from the ecological context. But in the end, we are all working on the same kind of problems, and in the end, we really should then try to find a place to meet, and where we then can actually then learn a lot from each other. And with these words, I really would like to stop. Thank you very much.